Welcome to the webinar this evening. My name is Jess, for those who haven't yet seen my face, and I am part of the PKD Australia team. So I would just like to start by acknowledging the traditional owners of the various lands on which we are meeting this evening and pay my respect to elders past and present. So tonight's webinar with Professor Melissa Little is about human pluripotent stem cell-derived kidney tissue. Melissa, over to you.
Thank you very much, and thank you to everyone who's online. I can't see you, but I can see the number of people, which is really great. It's really a great opportunity to be able to talk to you as the PKD Australia community. And I am just going to try sharing my presentation. There we are. Can we see that?
Yep,
Jess,
you
can
are
you see
good to
that?
go.
Okay, that's great. As introduced, my name's Melissa, and I'm a stem cell biologist, and I've actually worked in the field of kidney and kidney disease and kidney development all of my life. And I want to talk to you today about some work you may not be aware of, and that is looking at how you can recreate a model of a human kidney in the laboratory to better understand disease. And we hope one day to actually rebuild a kidney. But today I'm going to focus on telling you about kidney organoids, but I'll start with the kidney. I think most of you are aware that most people have two kidneys and maybe less aware that inside your kidney there are these very beautiful tubes we call nephrons, which filter the blood at one end and carry the urinary filtrate down and out of the kidney to the bladder. The reason we survive as humans is that each of our kidneys has around about 1 million of these tubes that are doing critical functions, cleaning your blood, rebalancing your fluid so that it regulates your blood pressure, it regulates your bone density and many other things. The key is that these wonderful structures that keep us alive, all of the nephrons that you have in your kidney actually form before you're born. And so for many years, we've looked in animal models at where the nephrons come from. And during development of the fetus, there is this, in red here, population of stem cells that can both recreate itself, but also, when given the right signal, form little tubular structures. That's the birth of one nephron. One end is going to connect to the tubes that will take ultimately the urine out of the kidney, and the other end will pull in the blood vessels. And eventually you get this nephron forming. And that process happens over and over and over again in humans until about the 3rd trimester of pregnancy, at which point you run out of stem cells and you don't make any more nephrons. So that's a problem for us after birth, because if you have a damaging kidney disease, such as polycystic kidney disease, you're not going to be able to replace those nephrons because there is no ability to make new nephrons after birth. So as somebody that studies development and disease in the kidney, we're really interested to ask, all right, can we use our understanding of how these nephrons form during development to actually start again? And this is only possible because of pluripotent stem cells. So you've probably heard the term stem cell used in many different contexts. So what is a pluripotent stem cell? So the term pluripotent means a cell able to give rise to any other cell type. And originally during development, the only cell that's pluripotent is the very inner mass, the inner cell mass of the embryo. But in the mid-2000s, two researchers, Shinya Yamanaka in Japan and Jamie Thompson in America, showed that the molecular cues that are able to take any cell back to that state of being completely open to turning into any other cell state, that is, the genes needed to make any cell pluripotent, could be defined. And they actually showed you can take an adult cell. It could be a skin cell, it could be a white blood cell, and you can actually take it back to this induced pluripotent state, which is now a stem cell population that you can grow in the laboratory forever, and you can actually turn it into any cell type you like. The challenge of turning a pluripotent stem cell into a kidney, however, is really very complex because the kidney is made up of more than 25 different types of cells arranged in a very specific way. So we still could use our understanding of how the embryo would normally pattern through mesoderm to eventually form the nephrons of the kidney. And this is my postdoc Minoru Takasato. In 2014, we published the first method to take a pluripotent stem cell all the way through all the steps of embryogenesis right up to make this tissue that is now a human stem cell derived model of the kidney. And we call these kidney organoids. So organoid as in little organs. So what we started with was some of these pluripotent stem cells, which we'll come back to who are we making pluripotent stem cells from in a minute. But after 4 weeks in the laboratory, what we have now is a little tissue. This is a couple of millimeters in diameter, and each of these structures with a white blob at the end is actually a nephron. So each nephron has a glomerulus, a proximal tubule, distal tubule, and this is the epithelium that connects it all together. So that's quite remarkable. Not only were we very impressed to find these nephrons connecting to each other and forming the different subsections, the proximal segment, the distal segment, and the glomeruli, we actually also showed that around these nephrons were the cells that make up the blood vessels. You can see them here in red. And the glomerulus that should filter the blood is making these very specialized cells, the podocytes, as you would expect in a kidney. In fact, this is a glomerulus inside one of these microtissues. And here you can see that very classic structure of a Bowman's capsule and some podocytes, and the vasculature should grow in here. And so we've got many glomeruli in an organoid. You've got 100 or 200 glomeruli that are actually patterning and segmenting into nephrons. So that's quite an amazing advance. That's almost 10 years old now. And what that means is if we start with a human pluripotent stem cell, we can make these kidney organoids, which is a model of a human kidney in a dish. What it also means is that you can gene edit those. So you can use gene editing to add a mutation or to create a tag, or you can actually start with a patient. And a patient with a genetic form of any type of disease, in our case, we're interested in kidney disease, we can make a pluripotent stem cell from a 2 ml blood sample or from a Now, we're really interested in using these kidney organoids made from human pluripotent stem cells all the way across this network. Am I still there? This is just saying that my
Yes.
internet.
You dropped out for 1 little second, but you're okay.
Okay, let me know if that keeps happening. What I want to focus on today is whether we can model diseases of the kidney using these. So I think this community is very well aware of what inherited kidney disease is. And of course, autosomal dominant polycystic kidney disease is the most common, one of the most common genetic diseases in man. But there are actually many inherited kidney diseases caused by different gene mutations. 1 in 15,000 children born has some genetic defect that's going to give them kidney disease. And for half of them, we won't get a genetic diagnosis. And even with a diagnosis, it's really not going to impact the options for treatment. This will still be transplantation or dialysis. So there really is a great need for the development of new treatments. Is a kidney organoid going to be useful here? Well, what's really important is that we can model the patient's own kidney. So as I said, we can typically, because I work in a children's hospital, we take small blood samples. We can then use gene editing to make mutants or correct mutations. we make the organoids, and then we have a look and see what it can tell us about the disease. But of course, you can't model a disease unless you have the right cells there. And what we've got is a very small model of a very young human kidney. And the diseases that you get depend upon the genes that are mutated and where those genes normally play a role. So I'm going to start with the glomerulus. There are many, many genes that can be mutated that are genes normally only made in the glomerulus, and they give you glomerular disease. And we're particularly good at making very beautiful glomeruli in our organoids. Here's a glomerulus with the arterioles and capillaries coming into the glomerulus, and around every blood vessel is this very specialized cell called a podocyte. We can create these in our organoids. They make the appropriate proteins that would normally sit here in the slit diaphragm to prevent large proteins moving forward. And they express the genes we want to be present much better than any other model of a human glomerulus. And we've used these to show that we can model diseases such as finished nephropathic syndrome, which is a mutation in this gene, NPHS1. And we've even found new genes, such as this one, NOS1AP, and shown using kidney organoids that this gene mutation is causing a disease. But what about cystic kidney disease? So to have a good model of polycystic kidney disease, whether that's autosomal dominant polycystic kidney disease or autosomal recessive polycystic kidney disease, and the sorts of cysts that arise in patients with cystic kidney disease vary depending upon whether you're autosomal dominant or autosomal recessive. In autosomal dominant, you get these cysts budding out all the way along the nephron, whereas in autosomal recessive polycystic kidney disease, it's very restricted to a dilation of the collecting ducts. So can we model these using kidney organoids? Well, in fact, there's now quite a lot of studies in the literature using our protocol or other protocols that have come since, where they have either knocked out a gene or made a patient line to model autosomal dominant polycystic kidney disease due to either PKD1 or PKD2 mutations, or autosomal recessive polycystic kidney disease. And the latter includes our own work, which I'm going to talk to you about now. So let's have a look at autosomal dominant polycystic kidney disease. I think we're getting to the point now where this can be modeled. And this is a study from Andrew McMahon's lab in the US, where they made literally thousands and thousands of tiny organoids, and they looked for this cyst formation in the dish as a measure of the defect. So what you can see is here's a normal organoid, and here's one of the PKD2 mutation, and here's one of the PKD1 mutation. They could then take these, make many, 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 many of them, put them into little wells, and then add drugs and identify some compounds that may actually reduce cyst formation. This is very early drug development, but it was evidence that you can use an organoid to find a compound that might actually treat ADPKD. I want to talk mostly now about autosomal recessive polycystic kidney disease. ARPKD is much rarer than ADPKD, 1 in 10,000 to 20,000 children. It is caused by a mutation in a different gene, PKHD1, and it's what we call autosomal recessive. The gene makes a protein called fibrocystin or polyductin. We really don't understand what this protein does. What we do know is that if you mutate both copies of this gene, you get autosomal recessive polycystic kidney disease. So here's mum and dad. Mum is carrying a mutation. Dad's also carrying a mutation, but they're perfectly fine. But 1 in 4 statistically of their children may inherit both the bad copies, and this child would present with autosomal recessive polycystic kidney disease. Now, this is quite a devastating cystic kidney disease, and it can actually develop and be detected in utero for very early onset ARPKD based upon these large, bright polycystic kidneys on scan. The majority of these patients are actually not really neonatal, but there's a fair percentage that are presenting quite early in life, and the life expectancy is quite low. In autosomal recessive polycystic kidney disease, well, there's actually quite a lot of laboratories. I just pause for a second. Can you hear me still?
Yeah, it just cut out for a moment, but you're still there.
There's quite a lot of laboratories now that are developing models to make this the collecting duct part of the kidney. We actually found that when we take our organoids, we can take out the distal tubules of the nephrons and then change their media conditions and make these little trees that are collecting ducts in the dish. So we actually showed from a kidney organoid, we could take out the cells that we wanted, we could grow them in a certain media condition, and we could end up over 20 days making these sort of cauliflowered structures that, if we change the growth media again, move from being an early collecting duct tip to being a branch or a stalk, which is what we expect is the target cell that is affected by PKHD1 mutation. So we decided to model this, and we used gene editing, which I won't go into today, but is actually a very rapid way of being able to change a gene within the genome of a cell. We actually knocked out this PKHD1 gene. So we have lines that are normal and lines that have no PKHD1. We grew organoids from them, isolated the red cells, which is the epithelium that we want. We cultured them in the right media. We made these little cauliflowers, and then we put them into the media that turns them into the stalk or to the trunk. And what we see is that in the presence of a mutation in PKHD1, you can see these tubes, instead of being thin and tight, are actually dilating. But what about in a patient? Can you do this in a patient? So we have been collecting patients with ARPKD that present to Royal Children's Hospital. When a patient presents with renal failure like this or evidence of renal disease, we do genome sequencing. We then know exactly what the mutation is, and we can make a stem cell line from the patient. And in this case, we can use gene editing to fix one or both of those mutations in the laboratory and then have a look at the modeling. And so here, for example, is a patient line that has two mutations in the same gene. And when we do this culture, we end up with cystic, dilated structures in the dish, in comparison to the same patient where we've actually fixed or corrected one of the two mutations that are actually much tighter and smaller. So I'm going to summarize there, because this is quite a scientific presentation. I want to be able to open it up to questions, but we're actually advancing this now so that we can make more complicated and larger structures that are collecting duct, so that we can better study the molecular basis of cyst formation in patients with PKHD1 mutations. And we're also making them much smaller because cyst phenotype in patients. So just to show you that quickly, we're now going down and making tiny little tubular structures in wells with lots and lots of small wells inside them, so that we can make thousands of these tiny structures for drug screening. So with that, I'm going to thank my lab. There are many people in my group that work towards not just disease modeling, but actually rebuilding kidney tissue for one-day transplantation. But in particular, the work in the lab on PKD, and that has been supported in the past by PKD Australia, is driven by Sarah Howden, who runs all of our gene editing, has been worked on by Sarah Yue, who is right in the middle here, and is really supported by two clinicians at the Children's Hospital, Kathy Quinlan and Tom Forbes, who were also a part of that project. So I'm going to just stop there and answer some questions.
Amazing. Oh, there we go. Thank you so much. That was incredible. I guess while we have some questions coming in, so for everyone, if you have questions, please pop them in the Q&A. The question that we like to start off with is why you started researching kidneys in particular. Obviously, this is relevant to our PKD community. So yeah, what brought you to kidneys?
I have to admit that it was chance. When I went
Yeah.
to university, I actually think I thought I was going to be a zoologist, and then I discovered human physiology, and it was so fascinating to me. And my first research project was in the kidney, and it was looking at the renin-angiotensin system, which the kidney uses to regulate blood pressure. And then my first job as a graduate was in kidney cancer. And I worked for some years in a type of childhood kidney cancer where we were looking for the genetic basis. And so I just kept learning about the kidney and then wanted to understand how the kidney formed, how it develops, what the genes do, what happens when they go wrong, how kidney disease forms. And then ultimately we started looking at stem cells and how we could use them to help patients with kidney disease. So it's just something that I've remained fascinated in, that I came across by accident.
I love that. From zoology to nephrology, almost. That's
Yeah.
amazing. Um, I guess another question that had come through just on our when people registered was that could this work that you've done and with the organoids, if not being a replacement for a kidney, could it maybe just extend the life of a kidney?
Yes, and I could have spoken about this. When we first developed this protocol to make human kidney tissue in a dish, I never believed it was going to be able to treat a patient directly. But we're much more positive about that now. It's a very, very big task to make an organ the size of a kidney that has so many functional nephrons, but we're not even sure how many is enough. Even in humans, there's a very big range of nephron number, and what we're finding is very big amounts of tissue. Now the challenge is how to make it really well, and we are interested in whether we can deliver it back into a patient that's failing.
Mm-hmm.
Can you hear me still?
Yeah.
And we're in the process of doing animal studies just to answer that question. Our hope is that we will be able to one day develop stem cell derived replacement tissue, even if it simply keeps a patient off dialysis for a period of time.
Mm-hmm. I know that's the hope of a lot of our communities is delaying the dialysis. Um, Another question that I think that was on everybody's minds who registered was if you were to put a time frame to this, how far away could you speculate that this is close to happening to creating that kidney tissue for a patient?
Yeah, it takes a really, really long time to make a new therapy like this. If I ask, well,
Mm,
what can I compare this to? So we are starting to see pluripotent stem cell types going into patients. I lead a consortium worldwide that's working in this space, and one of our teams in Sweden is making dopaminergic neurons for Parkinson's patients, and that is now in clinical trial. So the first patients have received stem cell derived dopaminergic neurons. So that took 15 years. And this is just phase 1 clinical trial. So there's still phase 2, phase 3, manufacture, market valorization. There's a long pathway beyond this, but first in human, 15 years. So we're 10 years from actually starting this process. We're already putting tissue into animal models. I'm hoping that within the next 10 years, we will be in patients.
That is incredibly exciting.
Yeah,
Knowing from the researchers that we have heard from, knowing the depth and the breadth of science and time that it takes, I think a decade, that's nothing. That's
well,
amazing.
I'd like to still be around when we're actually doing this.
Yes.
So
And
yeah,
I will say that I do not mean to say that that's nothing flippantly, because I realize that timing is everything to people who also do not want to go on dialysis. But yeah,
yeah.
in the realm of science, the fact that it could be within our lifetime is an incredibly exciting thing.
Yeah, well, we're really trying hard. That's what I can say.
Yes. Another question that had come through, hopefully this can be answered. The question is this work, your work, helping understand the effect of GLP-1 on the kidneys?
Yeah, that's an interesting question. I would say at the moment not. So I think for those who don't know what GLP-1 is, this has now taken the diabetes world by storm and also the obesity world by storm. The GLP agonists appear to assist in stabilizing diabetes patients, but also reducing weight. Exactly what the long-term consequences are, we really don't know, but there is some risk of renal disease in diabetic patients who are on GLP-1, and that would be really quite amazing. The application of that to polycystic kidney disease, I think it's really too early to say. So, while I'm kind of working in the science fiction world of can you really take a stem cell and make a new tissue, it's really important that other approaches are moving along as well. And for example, just using these organoids to screen drugs, if we could better understand what's wrong in patients, what's causing cysts, and how we can reduce them or slow them down, that may be just as good as transplanting a stem cell tissue to give you some new nephrons. So we have to take all options on the table.
Yeah.
And is it worth putting GLP-1 on an organoid in a dish? I'm probably my biggest skeptic. I would have to be asking myself, which cell is GLP-1 targeting? And have I got a model that's the right model to use? But it's an interesting question.
It was, yes. Well, that was all questions on my list. If there are any questions from our attendees, please speak now or forever hold your peace. I'll give you a minute.
I'm also quite happy to answer questions if I lost anybody. I tried to make this fairly deliberately short so that people could ask questions, but if it was too confusing, I'm happy to ask to try and explain better.
I will ask just for clarity's sake. So it seemed that the majority of what you had progressed in the modeling was related to ARPKD.
Yes, it was.
Are you on a similar level for ADPKD?
So in fact, many people are working on ADPKD with this model. ADPKD is somewhat different because we're sort of cheating a bit when we model ADPKD, because with autosomal dominant, you inherit one mutant gene, and this is why the onset of the disease is so late, because the cysts form when a second mutation occurs. So to do that in a dish with a kidney organoid isn't going to work because you haven't got the time to wait for a second hit to form a cyst. So what they do is they artificially knock out both copies, and it does form cysts. And as I've shown you that while we're not working on it, there are groups in the US and in Japan that are screening drugs to find compounds that will limit cyst formation or cyst expansion. And we are collaborating a little bit with researchers in Monash University who think there may be a way of reducing cyst formation by knocking out another gene. How you actually do that in a patient is not impossible, but it's coming along.
That sounds similar to what at Monash, I think it's Professor Ian Smythe and Denny Cottle.
It's exactly.
That's it. Yes, we had them on
Yes.
our webinars earlier this year, and hearing what they're doing with gene editing was fascinating.
Yeah, so they are also developing a way of degrading a protein that they think if that protein is gone, you may be able to suppress cyst formation, which I think is very exciting work.
Mm-hmm. Another question, from just another practical point of view regarding clinical trials, if these organoids are grown successfully and treatments are found, does that mean that a clinical trial time frame would be cut down because treatment may have already been successful on the organoid, or does it just mean that it's ready to then progress to a clinical trial with people? Like, does that actually help or hinder the clinical trial process?
It certainly doesn't hinder it. I think that the kind of standard process of developing a new drug is you put a whole lot of drugs across cells in the laboratory that are not relevant. You know, they may not even be human. They could be dog kidney cells, or they could be marmosets, and so then you take those drugs and you move them through into mice, and mice aren't human either. And so it's a very long path. And the problem with that path is it's expensive, it's long, and you can still reach a phase 1 clinical trial and find that nothing you did in the laboratory is of relevance to a human, and or that compound is nephrotoxic in a human. By having an organoid, particularly if we find that a drug that already exists has an impact, then I think that should shorten the path to clinical trial substantially. I think we're also working on seeing if we can make these a good readout of whether a drug is going to damage the kidney, because there are many drugs that actually damage the kidney. And I think this may also be a way of preventing us putting drugs into patients in clinical trials and then finding out that there are side effects that affect the kidney.
Excellent. That's wonderful. Okay, well, no other questions have come through. So with that, I think we'll finish up the webinar here. I just wanted to thank you on behalf of PKD Australia for taking time out, as I understand you are a very busy person, and condensing all those years of work, the last decade of work, into a presentation for us this evening, and for answering all those questions that I had. So thank you everybody for attending, and Melissa, thank you so much for your time and sharing your expertise and experience. And
Thanks, it's been a pleasure. And I just want to congratulate PKD Australia for what you're doing. And thanks for giving me the opportunity to speak to everyone. Thank you.
Wonderful. Thank you. Good night.